today is going to be a little bit heavy lecture, okay? So bear with me. Today we're going to introduce lots of things that pertain to the animal kingdom. Are sponges animals? No? Who says yes, who says no? Sponges. Somebody says yes? Who says yes? Raise your hand. So who did not raise their hand says no or is neutral? Don't know. Do not know. Good, I'm glad. So it means that you're learning something in this class. If you already knew, why would you be here? So, in fact, they are animals, as strange as that can be. They don't move, but apparently they don't move. We'll see that there is some form that they produce and does move. Okay, so they do meet that condition that we learn about animals, that animals move. Animals move at least at in one stage of their life cycle. Okay, doesn't have to be the adult. So they live in the ocean. They don't look like any other animal you're familiar with, right? They don't look like animal at all. They don't have a head. They don't have eyes. How can we classify sponges like animals? Okay. And here is what we have to learn today. What are those features that we recognize as common denominators to classify animals? Otherwise, you wouldn't know where to place sponges whatsoever if you didn't look and you didn't know about these features that we're going to talk about today. Okay? It is a little bit tedious, bear with me, but some things are interesting nonetheless. Okay, so as I told you many times, this course focuses on the kingdom Animalia. What I have told you so far about fungi, plants, proteus, that's it. We're not going to touch on these groups anymore. Now we're going to focus on animals. So we have to understand what all these different forms of life here represented. Here are real pictures. have in common. I don't have a picture of a sponge here. I should add it actually. What is that makes all these classified as animals? Before we move, I should switch these two slides. I don't know why they're backwards. I want to do an exercise with you. I want you to write down the first animal that comes to your mind. Just that. You can do that for me, right? I hope. The first that comes to your mind. Tiffany, what did come to your mind?
Dog.
The dog. Perfect. Jamarian, what did come to your mind?
Black mamba. Snake.
Okay. Interesting. Hilario, what did you write?
A lion.
A lion. Perfect. Anna.
A red panda.
Red panda. Interesting. Israel.
A penguin.
Pink. If I kept asking, I guarantee you, probably maybe one student would give me in this class the name of an insect, if any. Most of you gave me names of vertebrates, okay, big animals. But I like to do this exercise because it introduces one important notion that you have to have. The most successful group of animals on Earth is insects, okay, as Google learned. 2/3 of all the animal species, 2/3 of all animal species on Earth are insects, okay? Yet nobody ever tells me, or some, maybe once or twice I had an insect, okay? So let's try to take this bias out of our mind that animals are a dog or a lion or a penguin, all these vertebrates, okay? Animals much more than that, and especially insects, okay? Now, this slide should be changed to the other one. I probably will do it. These are some of the characteristics that we need to know about animals. And of course, it's all written here, but we're going to kind of dissect all what these lines mean, and especially the last lines. Okay, I need to start explaining to you the word diploid. This word, diploid, is a word that you have to start learning if you don't know from school already. Anybody has an idea what diploid means?
That's two, whatever it is.
It's two or whatever it is. I like that. She's analyzing the word. Die, two of something. Perfect. Okay. Ploidy refers to the number of chromosomes, number of the sets of chromosomes that we have, or an animal or a plant has in their genome, in their nucleus, you can say. So, as opposed to diploid, there is the word haploid. So, if diploid means double of something, haploid will mean half. Okay, these two words you must learn from me. So, refer to the number of sets of chromosomes. So, as animals, no offense, you guys have received one set of chromosomes from mom and one from dad, right? You know that. Whether you like it or not, you are the result of your mother and father chromosomes. So, and I shall say your grandparents mostly. But we'll learn about all this. So, you receive two sets, okay? It's kind of like looking at this in this view. It looks like evolution has created a system starting from genetics and going to features, anatomical features. Everything that is really important has a backup, even chromosomes. You have the identical genes, two identical genes, right? And of each representative in your body. It's kind of like maybe a backup. If one doesn't work, because maybe it's mutated, there is the other one. It comes into play, right? We have two arms, two eyes. Okay, all these important features are duplicated in our body. Okay, and so also chromosomes are. So being diploid means have two sets of chromosomes. Being haploid means have one set. Haploid refers to cutting in half the diploidy, basically. So we are diploid. We all agree, right?
Yes.
But do you think there is any type of cell in your body that is haploid? For some reason, there's only one set of chromosomes. There should be. What that could be?
Reproduction.
Sperm, egg, reproductive cells. Absolutely. Because when the next generation has to be produced, when you are going to mate with another individual of the same species, if in your gametes the number of chromosomes wouldn't go half, whenever the two gametes fuse together, you would double up, right? And at every generation that would happen. So we will end up with thousands of chromosomes, I don't know how many, like generations. That wouldn't be possible. No, even a small alteration in the chromosome number can bring to very deleterious consequences. Can you give me an example that you know of syndromes that Down syndrome? Down syndrome only has one small chromosome, and instead of being present twice, so instead of being diploid, it's triploid. So there are three representatives of only one chromosome, the number 21. Look what happens. Not only these people look different, but they have lots of problems, okay? And in most cases, most of the other chromosomes, excess number of chromosomes, other excess than 2, higher than 2 are not permissive for life. We don't even observe that. There is no syndrome associated with chromosome 1, 2, 3, 4. Why? Is that doesn't happen?
You would die.
It's miscarriage. Okay? So they happen equally, just like the chromosome 21. The chromosome 21, because it's a very small chromosome, allows for life to happen. Okay, so we learned the word diploid and the word haploid as well. So, with all that said, we know that all animals in general have an adult dominant diploid stage. Think about yourself, you're an animal. Again, no offense. And your dominant stage, most of the cells of your body are diploid, at the exception of just a small fraction, which is the reproductive cell. So that's what that word dominant means. The dominant portion of your body is diploid. Okay, so that's true for all animals. And that is a result of the fact that all animals, no matter what, reproduce sexually, right? Because sexual reproduction implies production of gametes that fuse together and establish diploidy, okay? Who knew that sponges can reproduce sexually? Otherwise, could they be animals? Yes, they do. We'll learn interesting things about sponges, okay? Sponges are so incredible. I just think about sponges all the time. And I watch SpongeBob with my kids, and I think about sponges even more. So, yes, they produce gametes. Although we'll see, they also have a plan B in terms of reproduction. We'll see. We'll start talking about sponges today already. All right, so we already learned that animals are motile. They move during at least one stage of their life cycle. Doesn't necessarily have to be the adult. So sponges, as you see the adult body, they don't move. But let's look at the life cycle of a sponge and let's see if there is any form in the life cycle that moves. Maybe that's the case, okay? All right, the other important thing we're gonna learn about today is the embryonic stages that characterize all animals. Going from that one fertilized cell that all of us were at the very beginning, day one of our life, right? Did you know there being a protist? Did you ever know you were a protist at some point? At the very beginning of your life, you were one cell. Even in that regard, you know, ontogenesis recapitulates phylogenesis, right? You were a protist the very beginning because you were just a fertilized egg. When the sperm came in and then the two nuclei fused together, say you established two sets of chromosomes here, this was you, okay? This was already you. How many pairs of chromosomes should I draw here? How many pairs of chromosomes should this cell have? Only 2? This is human. This is you. This is you when you were first born. Now when you came out of the body of your mother. This is you. In this cell that you were a protist here, right? Because you were unicellular, right there. Just a few hours, really, but you still were that. How many pairs of chromosomes do we have? 23, okay? I'm just representing 2 of, otherwise by the end of the day today, I'll still be drawing. So this is you, but then when after 9 months or so, you don't look like this, you look like this, crying. Oh, why did you take me out? What happened? What happened? Cell division. Wow, yes. This cell, after just a few hours, divides and becomes two cells. Each cell receives the same set of chromosomes, right? Because all the cells of your body must be representative of all this genetic material, okay? I'm not going to represent those chromosomes again. What's going to happen to these two cells? Each of them divides again. Mitosis, I hear. Very good. We're going to learn about mitosis. So after many cell divisions, this is who you become, right? Okay, now, today we're going to learn about milestone stages that are in the beginning of these cell divisions, okay? We're going to name them and describe them. You have to be able to describe them, even draw them for me, okay? So this is what we're going to learn about today, okay? So I'm skipping here, blastula, gastrula. I will explain it to you in a minute. The other important feature of animals, we already learned this, distinguish us from fungi. We have intracellular digestion, not extracellular. And think about an amoeba. You also are an amoeba as well. You're just like you're a protist here, genetic protist. Then there are some cells in your body that are amoebas. And finally, I found that video that shows a macrophage in a petri dish, a human macrophage chasing out bacteria and eating them just like an amoeba would do. So again, so an amoeba will ingest anything on its path and digest all what it ingests internally inside the cell. That's what our cells do too. Our cells eat all the time in our body, okay? And digest the food inside. Most of animals, we can recognize some kind of symmetry. We'll look at this symmetry in a minute as well. Okay, so these are main characteristics. What I haven't told you is that this very cell, this U that you were at the very beginning when after an egg cell had been fertilized by a sperm cell, is known as zygote. Okay, we'll learn this word also very well. So zygote. Okay. I don't know if anybody's thinking about something. It seems like a new being can be created by fusion of two gametes, an egg and the sperm here. What about if I fuse two eggs? What would that happen there? A new being could also be born because I'm fusing two haploid cells, two gametes, or two sperm cells, That's really possible. But do you think scientists have questions? Of course, they have questions. Scientists, they ask a question all the time, and they will do the experiment to answer. If I fuse two eggs, what would you expect that maybe I can create a new being?
So, if all this happens through fusion, what has to go wrong to be conjoined?
When you are asking when conjoined twins are born?
Yeah.
So, conjoined twins come out by a process where, what I didn't draw here, there is a membrane here, okay? And after the first division, okay, the two cells separate. They should separate in two, and most likely they have separated in conjoined twins. Somewhere along the way, so what happens here is that at this stage, there is a separation that appears where each of those two cells that have come up from the first division, for some reason, continue their path as an individual rather than these two be part of one individual. And today we're going to learn something. I'm glad you asked this question because we're going to learn something really interesting about embryos today. Okay? Hopefully you will think that it's interesting. What you can do with embryos and what these cells represent, up to at least 8 cells. Okay? Up to another division, you could still have basically believe it or not, each of these cells can become its own screaming poop factory, as I call my children.
You're terrible.
Okay? It's just a joke, okay? Each of that cell, even at 8 cell stage, can still become one individual. And I don't know if you know, there are just twins, a pair of twins, identical twins. There's very rarely 4 twins. You can Google it, they'll show you the pictures. They have happened naturally. Okay, 4 identical twins. Now, to answer your question, there is all kind of possibilities there where along the way, where these two, remember these two are very close, these 2 embryos, at some point during the development down the road, something has happened to where that membrane is broken and then the 2 have somehow fused together. There is no telling when this could have happened. Of course, you know, it's variable for each case. Okay, so, but probably much down the road than here. Okay, otherwise, I think here, if it happened very early, I think if it happened very early, most likely it won't be scared. It has to happen a little bit later in development in order for, you know, and to individuals, to conjoined twins to be alive, let's say. Okay, so interesting, very interesting stuff. But let's try to focus on what we're saying. We're going to talk about embryos pretty soon, okay? We'll give this. So before talking about embryos, let's look at several characteristics of animals. As I told you, most animals have some kind of symmetry. And it seems like the winner type of symmetry in evolution is a bilateral symmetry, the type of symmetry that we have. For some reason, when it appeared, it functioned so well that most of animals have conserved it. We can also recognize a radial symmetry. We're going to define it pretty soon. And there is one group of animals that have no symmetry at all, and these are the sponges. You cannot recognize any symmetry in sponges. This is a radial symmetry. This is a bilateral symmetry. We define them as, and these are just examples, bilateral, radial, asymmetry, no symmetry, right? Oh, yeah. We define, sorry, you must know this, the radial as a symmetry about a point, and the bilateral as a symmetry about a plane. These are represented here. This is about a point, and this is about a plane. The plane will divide the animal into specular images, right? Those are definitions, but visually you understand exactly what that means. Here, the slices are pretty much identical, the various slices. All right, so these are examples. The other thing we have to try to familiarize is the views of an animal symmetry. Dorsal, ventral, anterior, where the front of the head is, and posterior, where the tail is. Okay? I don't have a tail. What's my posterior? I'm not gonna touch it, but the rear. So dorsal, remember when you, as we are upright, so if you don't want to get confused, imagine that you are positioned this way. Students are not going to see what I'm talking about, but like a dog, and the dorsal will be here, ventral here, anterior and posterior, okay? And then lateral. Pretty easy for most of animals, probably except that for us. These are the views, okay? So let's now start focusing on the embryo and the milestone embryonic stages that we have to know of that are common to all animals, including sponges, okay? This is one of the points upon which we base classification and definition of animals. One of the main points. And again, how can we classify sponges and animals? I will show you pretty soon here. Life cycle of a sponge shows that in the early embryonic stages is exactly like you. Okay? All right, so after fertilization of two gametes, you have the formation of a fertilized cell, which is named really zygote properly. The zygote grows to maturity by cell divisions known as mitosis. We'll talk about mitosis when the chapter comes, the cell division chapter. Start familiarized with this word already. And during this mitosis, I depicted here very simplistically. Let's see, as down the road, as we go down the road, we can recognize several stages, okay, that we name blastula, gastrula, some of them. And larval stage is a stage that not all animals have. Do we have a larval stage that you know of? I don't think so. For instance, sponges do have a larval stage. It's an intermediate stage between the embryo and the adult. Okay, the changes really dramatically. All right, so this is a picture of the main milestone stages during the embryonic development of an animal. So we already talked about the fertilized egg, the zygote, that soon enough will divide or undergo mitosis to become two cells. So we call it two-cell stage, right? These two cells, each of them divides to become four cells. So we call it four-cell stage. Each of these four cells divides to become 8 cells, right? And I said embryo, and so on. So after 8 is 16, and then 32, 64. Then you have a morula. Morula is already between 64 and 100 cells. It's a ball of cells. Morula. Who speaks Spanish? In here. Mora. What's a mora?
Oh, okay.
Very good. A blackberry. Is that a blackberry over there? Is that a blackberry? Morula, from Latin, okay? So it resembles a blackberry. Have you ever eaten a blackberry? I think so. Is it filled inside? Is it filled core or is hollow?
It's the juice.
Well, depends if you can see.
Yeah, it depends on how they cook it.
How they pick it. Okay, let's say not so ripe, maybe. It's kind of filled inside, right? So that's the morula. As a filled core, if I sliced this embryo, it will have lots of cells inside. Okay? Then, so this is a blackberry. So you are a blackberry at some stage of your embryonic development. And then, after that stage, the cells that are inside in the core, they migrate out, and then you become a raspberry, right? This is hollow. So you're blackberry and then raspberry. The blackberry is morula. In fact, even in Spanish, it's mora. When you become a raspberry, you're named blastula, okay? So that's when you are a raspberry. You and the sponge. You both look the same here, okay? Then the next step is to become a so-called gastrula. So this hollow ball, I wish I had with me like a deflated volleyball, and I could show you. imagine a deflated volleyball. You can actually, and I can punch it kind of with this. I can make an invagination, okay, in that ball because it's soft. So when that happens, this is the initial formation of your gut, of a gut, okay? So when that imagination appears, that's the gastrula, okay? With an opening. So there is kind of like an opening and a sac, okay, here. At that stage, you are a gastrula, okay? So the three new words that we have to remember here. Obviously, I'm not gonna have to remember 2 cell stage, 4 cell stage, 8 cell stage because they're just pretty straightforward. There are the morula, which is a raspberry, the blastula, which is, the morula, which is a blackberry, the blastula, which is a raspberry, and the gastrula, which is when this movement happens to where a gut cavity is formed, okay? So these names you have to know for me, and even ask you to draw this thing for me. Is that hard? The blackberry, the raspberry, and the gastrula, okay? All right, so here's another picture. Again, same thing, 2 cell for stage, 8 cell stage. Here it puts another stage, 16 cell stage. We don't call it anything, okay? Then you have the morula, shown here. Then you have a blastula. In a cross section, you see that it's hollow. And then you have the gastrula. Remember, the gastrula has this opening, this invagination has happened with an opening. Now, that opening has a name, blastopore. It's a pore, right? Blastopore. And at this stage, also we have to name 3 important layers of cells. There are only 3 layers of cells at this stage. This is still very early stage of an embryo, okay? We recognize 3 major layers of cell. One that is outside, the prefix for outside is ecto, so we call it ectoderm. One layer that surrounds that internal invagination that has happened. So inside endoderm. And then there is something in between here, in between mesoderm. So ectoderm, endoderm, and mesoderm will be here, okay? Now, I shall tell you one thing. Most animals will have these 3 layers, but the very first groups that we're going to look at, such as sponges and cnidarians, in reality, they never develop a real mesoderm. And what happens is that in this in-between layer, they develop a gelatin. There are no cells in there. It's a gelatinous material. That's one of the differences. Remember, there's always some passage. There's always the first organisms of a group. They may be a little bit different than the following ones, okay? So this is a 3-layer embryo, right? 3-layer embryo, ectoderm, endoderm, and mesoderm, okay? As we will see, sponges are called 2-layer embryos because that mesoderm is not really a mesoderm, doesn't have cells, it just stays as a gelatin, okay? I want to tell you this little difference, but really the embryos look very similar to all the animal world at this stage. So if I encounter an organism tomorrow and discover a new organism, it looks strange. I've never seen something like that before. I don't know how to classify it. If it has these stages during embryonic development, I'll put it into the animal kingdom, okay? Plants don't have this whatsoever. It's kind of development. Very, very different, okay? All right, these are real pictures. You see how this imagination here is very, very clear. Okay, did not happen here yet. Okay, and as you go, A, B, C, D, E, and F. So as they happen, these are real pictures, okay? I think this is electron microscopy, is much more sophisticated microscopy than what we have done here, okay? So this is the gastrula with the invagination. You even see the cells here, the mesodermal cells.
Thank you.
I can tell you this is not going to be a sponge. I don't know honestly what animal this is, could be human, but I can tell you for sure it's not a sponge because I can see cells here. Sponge will not have the cells in here, will have just the matrix. Okay, the gelatinous material. Okay, oops. One question. Ecto, endo, meso. If I were to ask you to guess, take an educated guess, which tissues come out or organs come out from ectoderm, from endoderm, and from mesoderm? Think about your body. What's outside your body? So ectoderm, you would guess, gives rise to skin. What about endoderm? Endo.
Veins.
Internal
Internal organs,
organs.
okay, digestive, respiratory. What's in between our guts and our skin?
Tissue.
Muscles, bone, okay? Very good, so you already know it, okay? And in fact, I have here a list, okay? Now, the ectoderm gives rise also to nerve tissue, not just epidermis, okay? Mesoderm develops into muscle tissue, connective tissue, also blood, okay? Skeletal tissue, and endoderm. Endoderm forms all the internal organs, but also the layer of cells that surrounds our internal organs. It's kind of like a skin on our organs, let's say. That skin does not come from ectoderm, comes also from endoderm. Makes sense, right? It's still internal. So the epithelial lining of multiple internal systems comes from endoderm as well. Okay? So you already know all this, you can guess it. Now, here comes something very tedious, yet very interesting. So, there are two groups of animals. All the animals that have been identified, we can split them in two groups. One group is called protostomes, and the other is called deuterostomes, which the words from Greek mean. Protostomes means first mouth. Stoma is always an opening a mouth. Deuterostome is second mouth. Now, before going into the tedious detail of all this, I want to ask somebody, behavior. Think about a 10-year-old. If I ask my 10-year-old, what's the difference between these are two embryos at 8 cells each. This is an embryo of a protostome. This is an embryo of a deuterostome. You look at these pictures and tell me what you see. Are there differences there? And describe them.
Yeah, I think there's a difference, like the top. It's kind of different. And on the left one, they're kind of smaller, and the right, they're bigger. And from there, I don't see any difference.
Within the same embryo, what could you describe in deuterostomes and in protostomes, within the protostome and deuterostome embryo? There are differences, right, that you can describe. Liam, you want to help? What would you say?
just visual differences
Yeah.
between the two cells at the top? I mean, the one on the left, the top 4 cells look smaller than the ones on the right.
So we can say that the thingies of which these two balls are made, if I ask my kid, he would say thingies, right? He can call them cells because he doesn't know. You say the thingies of that ball are different sizes within the same ball, right? While the thingies over there are the same size. They look equal, right? That's a very important difference, because in that kind of embryo, which could be, for instance, embryo of an insect, okay? While this embryo could be a human embryo, okay? We are deuterostomes. Insects are protostomes. If I take away, listen carefully. I don't know if anybody in this room was born with in vitro fertilization. I don't want to know that, but if you are, this is very interesting. I can take these cells away from this embryo and still implant the embryo missing of 1/8 of it, right? 1/8 of the embryo is taken away, can implant into the mother's body, and screaming baby, perfectly screaming, will come out. So if any of you in this room knows that they were born with in vitro fertilization, and the parents chose to undergo pre-implantation analysis, which means you take away a cell from the embryo before implanting it to carry out analysis, for instance, for chromosomal abnormalities, I'm sorry to say you were deprived of an eighth of your being. But you still look normal, right? And so the people that were born after this procedure, they're still in their 30s, and very few. Statistically, we don't have many people born like that, and they seem to be fine, but I'm always questioning. Let's see what happens. If anything, in terms of, you have to consider so many things that could happen if you deprive of 1/8 of the body of an embryo. Sorry, I forgot to continue silence my phone. Of an embryo, such a significant part, right, of the body of that embryo. If anything could happen in the future, premature aging or disease susceptibility, you will have to have a lot of statistics to understand if there is any consequence. Anyways, long story short, we do have something more than insects apparently, because if I take away from an insect any of these cells that look unequal, the embryo won't develop, it will die. Okay? And so this difference, let's name these differences. We say that these cells here in the deuterostomes, which we are, have an indeterminate fate, have not decided what they want to be yet. They can be anything. In fact, as I told you, they could even give rise to perfectly single individuals. It has never happened naturally, but theoretically you could create 8 clones still. So we say that state of these cells is indeterminate. Okay, they have an indeterminate fate. These cells, they can still become everything, even an entire organism all over. Okay, while these cells are determinate. They're already determined to be a portion of the body of the new animal. So you take away a cell, you take away a portion of the body of the animal. Okay, that's what that means. Alright, so this is very interesting, but the more boring part that I'm going to tell you right now is this, which makes us name the 2 different types of embryos, protostomes and deuterostomes, mouth first or mouth second. Okay, so let's start from the deuterostome. This is us. Remember that blastopore that I talked about a minute ago in the gastrula. And this is the invagination that will become the digestive system. For some reason, you would think that the mouth develops before the anus. Yeah, that's how it started. But for some reason, things have shifted around during evolution where the blastopore, instead of becoming the mouth, will become the anus. And then another opening will appear on the embryo, a second opening, which will become the mouth. You have two openings, right? I hate to say it, but what you are is a digestive system with two openings. That's who you are. I'm sorry, I'm telling you terrible things today. First I started with saying you're proteus, then I went a little bit nicer. I said you're raspberry, blueberry, now I'm telling you you're a digestive system with two openings. But that's what this is, right? Digestive system surrounded by some sort of cells with two openings, okay? And that's who you keep being. And so the first in deuterostomes, the mouth is second. The first opening, the blastopore, becomes the anus, and the mouth appears as second. In protostomes, that's how evolution has started. The blastopore is the mouth, and the second opening that appears on the embryo becomes the anus. So mouth first. The first opening, the blastopore, will become the mouth. Here is the second opening that becomes the mouth. And that's what protostome and deuterostome means. Remember, protostome means mouth first, deuterostome, mouth second. Okay? Or second mouth, however you want to say. Okay, so this was the most tedious thing, okay? I promise. But it does have some interesting consequences, okay? Now, keep in mind one thing. Your study guide tells you all these names, all these things, I think in a very dispersive way. The way I present it, I put it in a very systematic way, otherwise you get lost. But all what I'm presenting, it seems like it's different than the study guide. It's not. It's the same information. It just presented more in a more organic way, more systematic way than just telling you things in bits and pieces, okay? All right, so we need to know the 3 embryonic stages. We need to know the difference between protostomes and deuterostomes, who they are in general as group of animals, okay? This was what I was trying to tell you about the pre-implantation analysis that are done many times when you undergo in vitro fertilization. Okay? I had a student in my other class, she was commenting that she did this. She had to undergo in vitro fertilization. Not everybody is as lucky who like to just have children naturally. Some people must undergo that if they want to have children. But you can choose if you want to do pre-implantation analysis. You don't have to do that. They just make you sign papers, waivers of responsibilities, things like that. And for instance, in Italy, this is not allowed. Because in Italy, we have the Pope, which says no. So, the labs in Italy, they do in vitro fertilization, they take the embryos, they send them to England, they do the pre-implantation analysis in England, and they send them back to Italy. All right, so anyways, you decide if you want to do this. So as I told you, even a sponge has these very milestone stages, okay? And okay, in this case, we're looking at a starfish, but I think I have a sponge later. Now, what I wanted to point out, whenever you see these cycles here, okay, you go from the adult that is diploid through a process of formation of gametes, this process known as meiosis, as we will learn. Formation of the gametes fuse, the gametes are haploid, they fuse, and they form the first diploid cell, which is called a zygote. zygote divides several times to become first a morula. Many times the authors don't put the morula stage there. It depends what they want to highlight. It's really whatever, you will find it all. Some put more stages in here, some they take the morula stage. Here is the blastula that is depicted, and then an early gastrula, very first invagination, and then the actual gastrula, okay? With the 3 layers of cells, it tells you ectoderm, endoderm, it is actually making them different colors, okay, and mesoderm. And these animals, just like sponges, for instance, have also an intermediate form that is known as larva. This larva many times, it has a life of its own, many times moves around, okay? And then the larva undergoes a very dramatic change called metamorphosis to become the adult. Not all animals have the larval stage, but all animals have these embryonic development milestone stages. Okay, so couple of more things, okay? Bear with me. The digestive system seems to be so important, so much so that it seems like everything revolves around that. But that's the truth. It's such an important feature, organ. It forms right away in the embryo, that invagination. It's the gut system, right? Now, in the animal kingdom, we can recognize 2 types of digestive systems, okay? One is more primitive and it's called incomplete. And later on in evolution, a complete digestive system appears, okay? We need to understand what these 2 definitions mean. An incomplete digestive system is a sac-like system, like this, okay? Where there is only one opening for food to go in and for waste to go out. Only one, okay? And in this regard, there are some groups of animals we cannot even classify as protostomes or deuterostomes, because what I told you about protostomes and deuterostomes implies that you have 2 openings there. So animals that have this sac-like, okay, digestive system, we cannot call them protostomes or deuterostomes. We say that they have an incomplete digestive system. Cannot even be classified as protostomes or deuterostomes. We'll see who they are, okay? A complete digestive system has 2 openings, okay? A mouth and an anus, okay? Mouth and anus for food to go in and waste to go out. And then you can be a protostome and deuterostome depending on where, if the blastopore became the mouth or not, please. Okay? So this is also very important, and as we go through the phyla of animals, I will tell you these animals have an incomplete digestive system. We'll watch videos of these animals, some kind of disgusting videos of animals that are eating and pooping from the same opening, okay? Basically. And the complete digestive system has the 2 openings separate. All right, last very tedious thing, I promise, okay? We need to know about the coelom. So if I take this flatworm here, we talk about flatworms, and I slice it up, how cruel that is. But I will notice that there is one cavity inside the body, which is the gut cavity, right? Everybody has a gut cavity. Remember that all animals develop into a gastrula, just that cavity there that forms first. If that's the case, if I am a flatworm and I have only one cavity in my body, I am a coelomate. Well, A means non, coelom, coelomate. Now, if I take any of you and I slice you in two, it's a joke, I'll see something different than a flat worm. And what I see is the gut cavity. Of course, we'll learn about the gut cavity. Every animal has it. But I will see another cavity that surrounds the gut cavity, which is called the coelom. So we are coelomates because we have a second cavity besides the gut cavity, okay? And the second cavity, where all our visceral organs, this is a liver, are suspended, is filled up with liquid to kind of keep the organs in place, and is surrounded by a very important layer of cells that form a membrane called the peritoneum. Have you ever heard about peritonite? Some people get that inflamed, it's pretty dangerous. So, coelom is a second cavity that some organisms have evolved that surrounds the gut cavity and has also a membrane, a very defined membrane, a very defined layer of cells known as peritoneum. So we have acoelomates and coelomates, okay? Does evolution go abruptly from one form to another, or there is always, most cases, something in between? Some kind of always in between situation. Check one, check two.
Check two.
Check two. Yes, in fact, there is an in-between situation that is called pseudo-coelomate. Some organisms, we'll talk about these worms in particular, are a passage where we can see there that in evolution a coelom has appeared. So if we slice them, we see this cavity that surrounds the gut, but the cavity is not surrounded by a very defined layer of cells. There is no peritoneum. So it's kind of like considered in between, almost there but not there. So that's why these organisms, these are specific worms we're going to talk about. Very nasty, some of them friendly. Pseudo-coelomate. They're defined because they're pseudo-coelom. Okay? All right. So the very tedious part, I think, is done. Okay? Again, the 3 milestone stages of embryonic development to be known. Okay? Try to depict them and make drawings. I think it's very difficult. Protostomes and deuterostomes. And the definitions that we talked about today, diploidy, haploidy, what's a diploid organism, what's a haploid cell. Coelom is another one. And what does it mean to have a incomplete versus complete digestive system? You literally could make these drawings for me. I don't care, it's a schematic thing, okay? This is a digestive system, okay? An opening for intake of food, another for to expel waste. All right, just a couple more things. Many animals have body segmentation, which refers to repeating series of body units. They can be identical or non-identical, as we will see in some organisms, such as earthworms. They appear to be identical, repetitive segments, while in insects, these segments are unequal and they're more specialized, as we will see. Okay. I'm gonna finish this a little bit early, okay? Because I know you guys are getting tired, but I didn't want to interrupt. I'm gonna take in one breath all these definitions, okay? So, now, as I told you, we're going to zoom into the animal kingdom. We're going to touch on the main phyla of animals. So remember, we learned about domains, and we learned about eukarya, okay, domain. In the eukarya domain, we have to put sub-boxes there, which are the kingdoms, where in the kingdom Animalia, we're zooming in more, and separate these groups of animals based on very broad characteristics. Can I put a sponge and a human being just in the same box, Animalia, and close the box? Won't be fair, right? We kind of have to make distinctions to understand how evolution has progressed, right, from sponge to human. So we're going to look at the different phyla, the different main groups, and understand and define the main characteristics. We will be talking a lot about open and closed, complete and incomplete digestive system. We're going to talk about protostomy, deuterostomy, and it's not that difficult to remember if you remember the sequence of these groups that I'm going to show you right now. So basically, these are all the groups of animals that we're going to go through. We're going to start with invertebrates, because these are the first groups to evolve. And then we're going to end up with the chordates that include the vertebrates. When we get here, we're going to zoom more into the mammals, for sure, because we belong there, and kind of learn more characteristics. But it's easy, because this is where most of your knowledge is already about animals, okay? So we're going to start with sponges, okay, the first group, and then move on with cnidarians. Then we're going to look at flatworms, okay, learn some interesting things about flatworms, nematodes, mollusks, annelids, arthropods, which are insects, basically, echinoderms very shortly, and then we're going to zoom into the chordates. Who remember when we did the examples of classifications, I talked about chordata. Who remember what I said about chordata? We are in here. Why chordates, chordata are called such? Refers to chorda, cord, and a cord, a rope-like structure that evolves in the embryo. That could become a spine. In us, becomes a spine, but in some chordates, it does not become a spine. It stays as a gelatinous thingy, okay, as we will see. So let's look at this phylogenetic tree, try to understand what's happening here. First there was an ancestor here. This is a whole clade, right? I could say this is a whole clade, Animalia clade, right? There is one first ancestor, a lineage that splits in two. One lineage becomes the sponges, and it seems like there are no tissues there. Another lineage evolves tissues, okay? And of this lineage that evolves tissues, there is another split, and here it's about the symmetry, radial versus bilateral. Then there is another split, acoelomates versus coelomates. Now you understand how important it is to know these terms and what they mean, okay? Even classification is done on coelom, okay? Okay, so the first group of animals we're going to look at is sponges, okay? I have other pictures here, other phylogenetic trees. My favorite is this one. To me, it's more clear, okay? Feel free to look at this one or this one if you're more like artistic and you remember this one better. But the sequence here, if you remember the sequence for me, you'll know that all the groups that come before the pseudocoelomates are acoelomates, right? And all the groups that come after the pseudocoelomates are coelomates, right? Makes sense. That's the in-between group. So if you remember the sequence more or less, you'll know that the only group of pseudocoelomates are the nematodes, which we're going to talk about, and all the groups that come before are acoelomates, and the others that come after are coelomates. Okay? Try to kind of visualize. I'm gonna show that phylogenetic tree over and over. Okay, just a couple of minutes, and then we go. Okay. to introduce sponges, okay? The first group. Thousands of known species, all different, okay? And they have no symmetry whatsoever that you can recognize. They don't move, they're not motive, right? How can we classify them as animals? They don't move at the adult stage, but they produce a larva that moves around. So remember, definition of animal, animals move at least at one stage of their life cycle. So that condition is met. Sponges reproduce sexually, amazingly enough, although they have a plan B. They also reproduce asexually. This asexual reproduction is called budding, okay? And if I take a piece of a sponge, and I have videos to show you experiments they've done, I take a piece of a sponge and I put it somewhere else, that sponge will become a big body again. So that's asexual reproduction, budding. They can bud out pieces. They can bud out and form an entire sponge again. Sexual reproduction happens through the formation of gametes, okay? And sponges are hermaphroditic. Who knows this term, hermaphroditic?
They have two sexes.
They have two sexes. They are, let's say, they produce egg and sperm on the same body. If you define hermaphroditism this way, you include all organisms. Don't say they have both organs, female and male organs, because sponges don't have even tissues. You cannot say they have organs, okay? So they are hermaphroditic, but their hermaphroditism is alternative. So this sponge represented here, this summer is a boy, the next season will be a girl. It's alternative, right? So that's what it means. So the sponges are hermaphroditic, but they never produce egg and sperm on the same body at the same time. They alternate. They may produce egg one season and the next season will produce sperm. Why do you think that happens? Why wouldn't it be more convenient to produce egg and sperm on same body? Maybe fertilization will happen very easily because it's on the same body. Reproduction are more efficient.
Genetic diversification.
Genetic diversity, okay? To promote, it's an evolutionary strategy to promote diversity because the egg or sperm produced by this body of this species will need to meet with the egg or sperm of another sponge in order for a new individual to come out through sexual reproduction. Okay, yeah, I'm gonna stop here and next time we'll dedicate a lot to sponges.
You were like,